What is a genome?
Well, for a long time, the scientific community defined the genome as DNA that codes for proteins.
Proteins like steak and cheese?
No, not like steak and cheese. Proteins are molecules that have many important functions in the body, including roles in structure, metabolism, protection, regeneration, you name it.
So my genome codes for proteins then?
Well, not exactly. At a basic level, the genome is like a piece of music. At some point, each cell has one, and it's made up of nucleic acids like DNA or RNA, which are like the musical notes. But you need more than notes to make a song, just as you need more than nucleic acids to make a cell work. The whole genome of an organism can even be the combination of several different ones, like how humans have nuclear DNA, mitochondrial DNA, and even the gut microbiome. The interactions between all of these genomes thus contribute to different phenotypes and heritable features. So genomes are actually way more complex than initially anticipated.
Yeah, that's a lot to figure out. How is it all done?
Well, with the arduous efforts of consortiums like the Human Genome Project and advances in technologies. Scientists have, for instance, discovered many additional regions of our nuclear DNA that have functions beyond making proteins. From these regions, they've learned that the vast majority of this genome doesn't even code for proteins. This led to the discovery of a whole host of other important DNA sequences, including introns, repetitive sequences, transposons, and functional RNA coding regions.
So, if these regions don't make proteins, what do they do?
Well, introns are nucleotide sequences that are removed during mRNA processing. And some genes can actually code for several different proteins thanks to alternative splicing, in which introns play a critical role. Repetitive sequences are patterns of DNA that are copied multiple times throughout the genome. They play important roles too, and one repetitive sequence forms the telomeres, which are responsible for protecting your DNA from degradation and slowing aging. Transposons are DNA sequences that can jump around and change their position in the genome, and can even jump into genes or gene regulatory sequences and disrupt gene expression. Functional RNA sequences are DNA sequences from which a functional non-protein coding RNA is produced, such as miRNA, siRNA, and long non-coding RNAs, which have regulatory functions in gene expression. Here, the phenotype can change, but the DNA sequence remains the same. What's exciting is that researchers are still investigating their roles and discovering new information every day.
So, these sequences are all working together in each cell of my body?
Correct! And although each of your cells has the same genome, with some exceptions like blood cells, sperm, and eggs, the regulation of this information leads to the hundreds of different cell types in your body. In addition to the functional RNAs we mentioned, epigenetics adds another layer of complexity to gene expression. Here, a range of different mechanisms are used to turn genes on and off without modifying the inherent DNA sequences, and, like the genome, this information can be passed on as well. So, the genome does not just code for proteins, but is also comprised of and regulated by the expansive and diverse non-coding regions and epigenetic landscape. And that's just the beginning. Commensal microorganisms that reside, for example, in your gut, on your skin, and in a whole host of other places, help coordinate many biological processes. Their genomes come together to form the microbiome, which works in concert with your genome too. But that's for another day. So, the genome is like a piece of music, where the notes are the genetic code, but the rest of the sheet music, the tempo, dynamics, and the voicing, for example, make the music what it is.